Okay, so it's really good to be back here on the final day of this Faraday course, and again to have Keith Fox with us, who's Professor of Biological Sciences at Southampton University. He's now going to give us his second lecture. Keith is actually senior executive editor of nucleic acids research, so has a particular interest, and his whole research area is in the field of DNA and so forth. So we have a real expert on our hands on this topic for this morning, which is genetic engineering. How far should we play God? Thank you, Keith.
Thank you. Thank you, everybody, for coming back for a second session with me after yesterday afternoon. It's a tremendous privilege to be able to talk to you. The title seemed to get slightly confused as we go along as to whether the "go" in there should have a D on the end of it, and whether with God or not.
Yes, the website would be
That's just a clever title.
And in common with a few other speakers, that's my email address. If anybody wants to contact me about anything, I'm very happy to engage in dialogue with anybody. k.r.fox and then universitysutton.ac.uk. What I'm going to do in this session is to expand on some problems. Cheryl in the last session gave us some very good principles that we could work through, ways that we might think about bioethical issues. What I'm going to do in this session is to give you some bioethical problems, and not necessarily to give you the answers to those problems. You can work them through for yourself. Sometimes I will give you some pros and cons and ways in which people are thinking about them. But I want to address particular issues and just to raise the problems. Of course, we're able to do genetic engineering and genetic modification solely because of our knowledge of this wonderful molecule. And it just gives me an excuse for 2 minutes to tell you something about DNA, since it's my life's work, and I spend my time talking to students and postgraduates and other staff about it. DNA is our genetic material. It's found in not every cell, but almost every cell in our body, and it's exactly the same copy of DNA in every cell that has it. And it's slightly different from everybody else's, and different, of course, from different species, different organisms' DNA. But yet, many similarities. So there are many genes that we have that are very similar to genes that are present in chimpanzees and monkeys and pigs and hamsters and guinea pigs and bacteria. So there is a kind of unity throughout the biological realm of using this molecule DNA. We see its structure, this almost iconic structure of molecular biology, this right-handed double helix, lots of different ways of portraying that in terms of colours or sticks or balls. But it's the fundamental of the genetic material of life. And within humans, we have this DNA packaged into 23 different chromosomes. We have 2 copies of that in all of our somatic cells, 1 copy in our germ cells. This molecule is fascinating. It's actually a very, very narrow molecule. It's a few nanometers in diameter, and yet in length it is monstrously long. Each cell within your body has about 2 meters of DNA within that. And it's able to be packed into a cell that's just a few microns across because it is so narrow. It's rather like cotton, if you like. So that's not a very good example. Something that can be immensely long but very, very narrow, and you can therefore put it into that tiny space. And we know quite a lot about it now. We've sequenced the human genome and sequenced the genomes of many other species. So we know the fundamentals of what drives them. We can begin to pick out particular proteins, particular control features, what is unique about humans, and there's probably not a lot that is in our genetic material, what is different about humans from other species. I put this up partly not to blind you with chemistry, which is one of my former disciplines, but to emphasize that in talking about DNA, we are actually just talking about a simple one. It's a polymer. It's a chemical. If I was to put in front of you a tube that contained DNA, you actually wouldn't be able to tell the difference between that and the standard piece of tissue paper that's gone accidentally through the washing machine and got dried at the end of it all, just looking like a white pulp. That's what DNA looks like. It's just a white, it's a colourless, despite the representations we put, it's a colourless molecule. It's just another chemical, very high molecular weight chemical, very big, very repetitive in its structure, but it is just a chemical. It's only when it's put in the context of a cell, it has some very special properties in what the cell then does with that DNA. Important to realise that the genetic code, the DNA, and it's been picked up by other speakers, is universal. The way in which DNA is read is exactly the same for us as it is for a bacterium. It is the same molecule in terms of its chemical constituents. The coding for how that DNA is then translated through messenger RNA through into protein is exactly the same. And that actually has some very profound things in terms of the origin of life. It says, I think, that all life as we know it originated from a single source. It doesn't necessarily mean to say there weren't other forms of life around that tried different genetic codes, but if they did not survive. All life as we know it uses the same genetic code. And because of that, it's therefore possible to transfer bits of DNA from one species to another one. As far as the organism is concerned, it's just a part of that big polymer that can be replicated, can be translated. There may be some problems with translation occasionally if you get the wrong control sequences. But basically, any cell will take any piece of DNA as just part of that big polymer. And so we can move it around from one cell to another. And that's the basis of the whole of this discipline of genetic engineering, genetic modification, is taking either something that's in an E. coli cell and modifying it, mutating it and leaving it in E. coli. That's fairly a benign sort of operation. Or taking something from a human and expressing it in a bacterium. Or taking something from a pig and putting it in a human, or vice versa. Being able to move that genetic material around from one organism to another one. I'll give you just a little bit of history of genetic engineering, going back nearly 40 years now. In 1972, and that was actually just before I started doing my biochemistry degree, so it's within my lifetime, was the first occasion of when recombinant DNA was used. That was actually transferring DNA from a bacterium to a bacterium. The first time when it was put into E. coli, a synthetic piece of DNA, 1973. Very shortly after that, the scientific community did begin to get concerned about what they were doing. And in 1974, questions started to be raised. What would happen if we accidentally took an E. coli, most of which are not the terrible pathogens that you read about in the newspaper, but are benign organisms that live in your gut and mine, and some strains of that are highly pathogenic and cause very nasty diseases. What would happen if we took one of those natural organisms that's there on your skin, in your gut, and actually there's 10 times as many of them on a cell count as there are of you in your body? What would happen if one of those somehow mutated by virtue of genetic modification and say we made it so that it formed cancers? Or it gave some horrible kind of disease that could spread throughout the whole of the world. So in 1973, 74 and 75, a lot of the scientists doing those genetic modifications drew breath. And we read these sorts of quotations in journals like Science. There is serious concern that some of these artificial recombinant DNA molecules could prove biologically hazardous. And the word hazard or risk is actually quite important in this. It's not actually whether it's ethically right or wrong, but is there a risk that's involved with this? And that really culminated in 1975 in this the conference that was held where scientists laid down some ground rules for how genetic experiments, genetic modification should be done. And it was described as a landmark of social responsibility and self-governance by scientists. They themselves, without any pressure necessarily from society, said, let's stop and think about what we're doing. It proved very quickly that most of those extreme fears were unfounded, but there are still constraints on what we can and we cannot do, or rather the confines in which you can do anything, as to what sort of level of containment any genetically modified experiments in the first instance have to be done. The first genetically modified species first was in 1976. Actually, animals were modified earlier, and in 1983 it's slightly technically more difficult in the first instance, and not anymore, to produce genetically modified plants. So this is a field that's developed over 30 years, and now the possibilities are immense. As we do this, there are some ethical principles that pick up a little bit on what Cheryl was saying, of why should we do this, what are the implications. And especially as we think about modifying animals, very few people get concerned about modifying bacteria, but they do get concerned about modifying animals or humans. But general ethical principles are some of those that are common to those found in medicine. We're doing it because we want to do good. There needs to be a benefit to society or to the individual. And weighed against those, of course, we don't want to do harm. Now, the medics know that those two are often in opposition to each other. In order to give a therapy that treats a disease, often the drug that's used will cause some harm. And the more severe the disease, sometimes the greater risk you can take in terms of allowing side effects. And those two have to be balanced against each other. A trivial disease would not be treated with a life-threatening drug. The similar principle applies as we're doing genetic modification. The principle of autonomy is also quite important. That of being able to make your own free decisions. And that applies to the animal, we'll say, being modified as much as it does to the human as well. And we'll come back to that one. And the principles of justice, of making sure that we're being fair to people. Maybe that's people with diseases, with particular needs, and being fair and just to the world in which we live. But of course people ask the question, isn't this beginning to play God? Wasn't it? Is that in the realm of life, is this God's domain and nobody else's? And in which we're able to move things around. And you see these sorts of photographs of a mouse with actually a synthetic ear being grown on its back. That's a rather extreme example. But people ask these sorts of questions. That, by the way, is not a scientific article. That's an advertisement, as it says in the small print, that was put there by those who were opposed to genetic modification. So we get this whole area of, are we playing at God in doing this? Are there boundaries that we shouldn't cross? This phrase itself is very emotive because the very phrase suggests that we're playing. It's something we do for fun rather than for a particular purpose. It almost suggests that we're not being serious about it. It's something that's a kind of an amusement, which actually I'll show is not the case. And it also asks the questions about God. What does God do, and that we don't do? You might say that if we are created in the image of God, and we might debate, as we already have done, quite what that means, but God is creative. And if we're made in the image of God, then part of that is reflected on us too, to be creative. And as we do genetic modification, actually are we acting as people made in the image of God with some of his character endued on us as well? I'll just throw that out as an idea to you. But ultimately people are concerned with genetic modification. What's it doing? Here is a quote taken completely out of context, but it maybe emphasises some of the concerns. This is taken from George Orwell's book Animal Farm, where at the end the creatures outside look from pig to man and from man to pig and from pig to man again, but already it was impossible to say which was which. And that's obviously talking in political satire, not in genetic modification. But for those of you, particularly those in the UK, who are science fiction fans, particularly of Doctor Who, may have seen some of the more recent episodes last year of times when there were hybrids formed between Daleks and humans, or between pigs and humans. And the sorts of implications that rather trivial fun bit of television might have thrown up. So in this particular episode, when the pig slaves captured the humans, the Daleks did an intelligence scan on them. And the humans that were of high intelligence were then used for the final experiment. In other words, they were hybridized with the Daleks, whereas those that were of low intelligence were turned into pig slaves. And this came from the two episodes, the Daleks in Manhattan and the evolution of the Daleks. And that's the sort of thing I think people fear about. Are we blurring boundaries? Are there questions of who might benefit from any genetic modification? Are we going to be splitting people into two groups, the genetically enhanced and the non-enhanced, the intelligent and the not intelligent? I think you can see the sorts of issues that are thrown out from that. So overall, the sorts of issues that are raised within the whole GM scenario are risk, what might happen that could go terribly wrong, questions of what is natural. I've already raised the question of natural law. Are there problems of boundaries between species that ought not to be crossed? We raise the question, what does it mean to be human? I'm not going to touch on it, but there's all the question of actually who owns our biological, in other words, genomic information that is special and different for each one of that. Do we own it? Does my GP own it? Does a pharmaceutical company own it? Do the lawyers own it? Do the insurers own it? Who has the right of access to my genetic information? It comes on the question of what rights do animals have as we genetically modify mice or sheep or cattle. Nobody asks them, and do they have any rights that say we shouldn't do that? There's the question of utilitarianism. Are we just able to use the biological realm around us to our own ends, regardless of the cost to that? And it also raises the question that I'm not going to tackle at all, I think will be dealt with later on in the day, as to when exactly does life begin for humans? And to put those questions in a different way, just looking explicitly at GM, there are two questions, I think, that are more to do with risk, not necessarily to do with ethics by themselves. So what are the health risks associated with GM? And are there long-term effects on the environment? Those are risk-type factors. And then you've got lots more questions that tease out the other ones that I mentioned. Does it blur the distinction between species? What control should be put in place? Is there pain and suffering? What if we have animals that begin to display human traits? Are animals just commodities? What if we make an animal that has a higher intelligence than it normally would do because of any intervention we've done? And who will have access to any GM technologies? Is it just the rich and the rich ones in the West, or are they globally available? A rather depressing phrase, I think, that came from Professor Steve Jones, who was a geneticist at University College London, talking about DNA and the implications of that and the way in which we think through what we're going to do. He said, when it comes to medical research, any medical technology that works is very quickly accepted by the public. Ethicists may not like it, scientists may not like it, but the public, if they believe it works, they will accept it, and legislation will always follow. Ethics has always followed science, it's never led it, and I don't see any reason why genetics is going to be any different. Ethicists would love to tell geneticists what to do, but I'm afraid the geneticists are not going to listen. Now, Steve Jones is a perfectly reasonable person. He's not the one who goes out and does weird and wacky things, but that was, I think, his fairly depressing statement, that actually we're running hard to catch up with the facts and the discoveries, and basically people say if it works, we will use it. I hope that we'd be a little bit more reflective than that. Let's start with a question then, of genetic modification. What is natural? The Swiss constitution has part of it, requires respect for l'intégrité des organismes vivants, for all living life and the natural environment. That is not what Switzerland would naturally look like, with green, luscious fields, if it was left to its own devices. It looks nice like that because we as humans have modified it, not genetically, but we've gone in and changed the natural environment. The Cambridge colleges that were around here look very good, and there's many of them have been like it for hundreds of years, but that's not natural. This is Fenland. This should be swamp out here. We should be probably under, at least walking around in marsh, if not under a few feet of water. That's not natural. So as we come to look at the biological realm, what is natural? There's lots of debates over GM and GM plants and vegetables. Here is corn. For a lot of, particularly in the US, corn is genetically modified, so as to be resistant to various herbicides, etc. The corn itself, the very plant, is not natural. It itself is a modified version of what is believed to be this sort of primitive popcorn on the left, which is found in Argentina. It's thought that that is the precursor of this, and that this one is selected for by various taking crops over generations and interbreeding them by traditional means, not by genetic modification, but you go from something that is pretty much inedible over here to something that has a very high market value and very high nutritional value. Similarly, the pigs that we might use for producing pork, they've got good muscle value, and yet it's now been argued that there's a single mutation in one of the genes that occurred a few hundred years ago that changes a single base in one intron of insulin-like growth factor 2 from a G to an A, and the net effect of that is increases the muscle mass of the pig, so of course it's much better for food, which is natural. This was done by accident. It's just a natural mutation that happened that we then picked up on and used to our benefit. But the natural pig out in the wild probably wouldn't be quite so muscular in terms of its meat content as the ones that we use. What about crossing the species barrier? Now, some people argue that we shouldn't do that it's unnatural, it's immoral, it's something that's in violation of God's laws. And that assumes that there are species boundaries that shouldn't be crossed. Someone might take the Genesis account, where God created creatures according to their kinds. Now, I think in the light of what Ernest Lucas told us yesterday, we might be able to put some cultural gloss onto that and say what that means. It just simply means, I think, that cows give birth to cows and sheep give birth to sheep. That's our common observation of the world around us. But to people who take this sort of argument, genes are seen as an essential part of an organism's being. It's telos, and it shouldn't actually be moved. It's fundamental to the organism. They see that genes are part of the complex web of life, if you like, almost that Gaia-type hypothesis. And they can't be treated in any sort of reductionist way without disturbing the balance of nature. And but of course, for those who take a view of evolution and say that actually species boundaries aren't fixed anyway, they've evolved, they've changed. What we're doing is maybe speeding up processes. We're doing things that wouldn't happen naturally, but there are no fixed boundaries between species that cannot be crossed over time. In fact, biologists struggle to define what they mean by species. Is it in terms of the ecology where it lives, its biological morphology, so the shape of it, its reproductive abilities, its evolutionary history? All these sorts of things define what a species is, and it's not a very simple task. Ask questions of what does it mean to be human. We could have a lot of lists, and you could add to this. Clearly we have higher cerebral activity, we're rational beings. We have a higher level of self-consciousness, we're morally aware, we have language, we have spirituality, we may be made in the image of God. Do any genetic experiments that we do start to blur these boundaries and actually either take away those from humans or add them to other animals? Some might argue that animals are not rational, and so they're not worthy of moral consideration in the way that humans are. So they can be treated as a means to an end. And I don't know if you probably don't agree with that, but others have argued that. But if you take that sort of view, What happens in other marginal cases? How would we view non-rational humans? Those who are severely disabled, with very compromised mental abilities. We would say, and most religious people would say, that they're of tremendous value just as themselves. And what we say about animals that display some capacity for rationality. You see, we're part of that continuum of the biological spectrum. We are mammals. We have about 30,000 genes, slightly less than that. 98.5% of our genome is similar to that of chimpanzees. Even 80% of our genes are the same as those in mice and in humans, and 60% of our genes are common with the fruit fly and the worm. There's lots that's not uniquely human. If we look at the size of our genome, and we dealt with this yesterday and the day before, when our genomes aren't particularly bigger than anybody else's, we've got about 3 billion base pairs in the human genome. Frogs have a similar sized genome, and the amoeba, that little single cell organism, actually has a genome that's about 200 times bigger than ours. It has a lot more what we might call junk DNA. So there's nothing special about us in terms of the size of our genome. There may be something special about the complexity of it. I'll come back to that in a moment. But we're also left with the problem that individual genes can produce more than one protein. When I was an undergraduate, we were told the dogma was one gene, one protein. And now we know that's not the case. A gene can produce many, many different kinds of proteins. And I'll explain a little bit about that in a moment. A gene can produce many different kinds of proteins in the same way a musical instrument can produce many different kinds of notes. And the way in which this is done is this may be a gene across here, which is split into regions that we call exons. They're the important parts. There's the bits that are expressed, and introns that are there in the gene. Sometimes we have no idea why they're there, and they're cut out. They're copied into the messenger RNA and then spliced. And then the functional protein, the messenger RNA coding for the functional protein, is put together. And you can see that this splicing pattern has many possibilities. So I've drawn that there in a case where black and red and green and yellow get spliced together. But in a different tissue, in a different circumstance, it might be spliced in a different way, so that we leave this green bit out. Or we might splice it a different way and leave the red bit out. So one gene can give rise to different sorts of proteins depending on which bits we take and which bits we don't take. And that's some of the tremendous complexity that's involved in the human genome. While we may only have something like 25,000 genes, which doesn't sound enough to create a human being, yet those 25,000 genes can result in the expression of far more proteins. So the process of splicing allows several proteins to be made from one gene, and actually it's been estimated that something like 15% of all genetic diseases arise actually for errors in this splicing mechanism, where the wrong bit is cut out, or a bit that should be cut out isn't cut out. And an example of that is the genetic disease beta thalassemia. So what I'm getting at is the idea that the concept of a gene is a very fluid one, and that there's almost nothing that we can say that defines a gene as being particularly human. What matters often is the timing of it, when a gene is switched on and when it's switched off. So if you look at a chimpanzee's head, a chimpanzee looks clearly different from us, but the genes involved in it are very, very similar in developing that. What it means, though, is they're switched on for different periods of time. So the jaws of a chimpanzee grow for longer than those of a human, whereas the cranium grows for a shorter period of time. The same genes, but switching, and where they're switched on and off very differently. There are all sorts of course, other genes involved as well. So within that, we can't often claim exclusive ownership for much of our DNA and our genes. We might use the phrase human gene and human DNA, but actually those are misleading terms. Often the gene is found in other organisms as well, and what's special about it is the timing of when it's expressed in the human. As I said before, DNA is just a chemical polymer. If there are any important human genes, maybe they're actually the master genes, the one that's control the timing, the switching on, the control genes, the repressor, the activator genes that take part in various stages in development, rather than necessarily saying this is a particularly human gene that marks us out as different. And I emphasise that so we must beware of genetic reductionism as we talk about GM. We are more than just genes. So what I call the fallacy of the gene for. There are genes for blue eyes, brown eyes. There are genes that dictate grey hair. There are genes that would dictate that I'm more likely to suffer from middle-age spread and obesity. But there is no such thing as a gene for homosexuality. There may be a series of genes that together interacting might give a predisposition in that way. Lots of things. There isn't an identity between one gene and one particular trait. And there is almost a fallacy of what I will call deification of DNA. So you can't tinker with it because it's so special. It's almost godlike. But we are more than just the sum of our genes. And sometimes in some of the arguments against genetic modifications, we are beginning to subscribe to a form of genetic determinism. I said about the genes and the orchestra. Can we say what is a human complement of genes as compared to, say, chimpanzees? And the analogy is one of the orchestra again. Is if you looked at an orchestra, you couldn't necessarily tell whether they were playing Beethoven or Bach or Stockhausen. It would be the same. You would, by looking at the instruments there, you wouldn't be able to tell what they were playing. You have to listen, observe what the music that's coming out of them. The instruments themselves don't define the piece of music that's being played. It is the score that's being read. So we mustn't, as somebody put it, confuse the violin with the symphony. Human dignity is attributed to the whole person, not just to parts of it, and there are very few, if any, genes that are specifically human. It's only a human gene in a human context. It's only a chimpanzee gene in a chimpanzee context. And that goes at levels above the gene as well, I think. Is there anything such as a human neuron, a human nerve cell? Probably not. It's a human nerve cell because it's there in a human body, and that nerve cell could be put in a mouse. In fact, I'll show you some examples of that in a moment, and it still functions as a very good nerve cell in the mouse. It's a human nerve because of the context in which we find it. So if there's that many similarities, how many genes could we transfer from one organism to another one? Does it matter transferring 1 or 5 or 50 or 500 genes from, say, a human genes into a mouse or maybe into a primate? And the answer when we come to that many genes is, to be honest, we don't know. You can transfer one gene and a human insulin gene expressed in a mouse doesn't make the mouse any different at all. But if we started to transfer hundreds of genes, we don't know. So proceed with caution is the ethicist's, I think, response to that. Don't rush in where there might be problems. So the production of creatures with overtly animal-human properties would create, I think, grave and complex ethical issues. And those experiments should only be done with extreme caution. And you might have a different interpretation on what extreme caution means to other people. Some people that means do it but be very careful, and for other people that means don't do it. What happens if we insert human genes into other animals? And that's been done plenty of times, and it doesn't change the nature of the animal. A mouse with a human gene is still a mouse. But there are plenty of safeguards that are talking about the things that we should and we shouldn't do. So the National Academy of Science recommend prohibiting creation of animal-human chimeras, where there's the introduction of human embryonic stem cells, and we'll learn more about stem cells later, into a non-human primate embryo. We start to blur human with chimpanzees. But they're less worried about chimeras such as a human and mouse, because there's such less in common between those two. and that the added to which no human embryonic cells should then be allowed to breed. These are experimental things used just for looking at cells within a laboratory. In fact, there was a project in which 100,000 human brain cells were injected into a fetal mouse brain. And that passed review. It was allowed to be done because even though 100,000 cells sounds like a lot of cells, it actually ends up with an organism that's still 99% mouse. And those nerve cells still function as if they were mouse cells. What if we produced a transgenic mouse with human genes, if it displayed any human characteristics, and how would we know? How are we going to... You can't ask the mouse, do you feel a little bit more human than you would have done if it didn't have the genes, and arguably we wouldn't know. I'll take a step aside for a moment and just to consider some of the social ways that genetic modification is looked at, and the way in which the media, particularly in this country, talk about genetic modification. And so they use very unhelpful phrases. And you see cartoons of frankenfoods and injecting dyes into all sorts of vegetables and organisms. You hear phrases like genetic pollution, frankenfoods, genetics, even that phrase the general public don't like. Transgenics suggests that something weird is going on. Mutation, horrible. Even genetic engineering is one that I think we have to be very careful how we use with the public, because engineers, with apologies if there's any engineers here, are usually involved in things like quality control, predictable outcomes, measurements, efficiency, utility, rather than the fun of life. Even genetic engineering suggests maybe something a little sinister. Added to which, of course, many people don't understand the science at all, so the phrases we use for describing that to the general public are extremely important, and not to give them the wrong impression. Let's think for a moment as to why we want to do any of these genetic modifications. We may want to do it for medical reasons, and I'll come to some in a moment. We may want to do it for research. Lots of transgenic animals are generated not because they themselves are going to be any real medical benefit, but because by producing them, we learn more about the ways in which organisms function. We knock out a gene in a mouse, or put a gene into a mouse, and actually see what does that do to its physiology, to its function. And by that we begin to learn about what causes organisms to function. There is curiosity, and we have to live with that. That's part of what it is to be a human, is to be curious, to ask questions. How do things function? Sometimes things are made by virtue of an ego. Right? The first person to clone a human may be incredibly unpopular, but they will also be incredibly famous. Right? There are commercial reasons for doing that, doing loads of these things, for generating new drugs, for generating new sources of revenue. And of course we might want to do it just for fun. Science is fun, and as long as it's done within the right boundaries, there's no reason why scientists shouldn't enjoy themselves in the process of doing that. We need to ask questions about who will benefit, the poor and the needy, or the rich and the powerful. So let me give you a few examples of some genetically modified organisms, and you can decide whether you think they're fun, whether they are for research purposes, or for curiosity, or for medical purposes. Here is Alba the bunny. This is a rabbit which has within its cells a protein that the gene for that came from a jellyfish, and it makes a protein that's green. It's used as a marker in lots of molecular biology experiments. In fact, I think a Nobel Prize was won recently based on the discovery of that gene and its use. And Alba the bunny, to all intents and purposes, is a bunny. But when put under a fluorescent light, it glows green because it has inserted within its cells that green fluorescent protein. And Eduardo Keck got hold of this, and he's used that, if you like, as an art form. So he says, my intention is to shift the discussion around transgenics and objects that are used for a purpose to an appreciation of the perceptual and cognitive life of that individual transgenic animal. The point is then to highlight the uniqueness of that individual, and also to shed some light on the question of purity. Here is a transgenic animal that challenges the notion of purity. So it's using transgenics just to ask a question. Here we have some genetically modified pigs. These are pigs that are modified so that they make a better use of phosphates, and so that there's less pollution in the environment as a result of pig farming. It could be used to reduce the costs of raising pigs, therefore to lower the food price to the poor. These are still look like a pig, they smell like a pig, and the meat tastes like pork. There are issues within that. Could the DNA that's been used in that jump to another species? And if it did, would that cause any problems? Almost certainly not. Does it affect the pigs? And it appears it doesn't. Might the low levels of this gene that's been introduced to the pig in some cases be toxic to some people or allergenic to others? And that is a question that might need to be addressed, is that they might be harmless for the majority of people, but could produce a protein that might produce an allergic response to a very small number. And we need to think in terms of not just the majority, but what effect anything we do has on a minority, and what we do to protect them. And ultimately, for those who eat pork, would you actually eat the bacon? Glowfish. Here's I keep tropical fish, or at least well, I did. I stole a fish tank, but it's not very well looked after, I have to say. These were produced with fluorescent genes within them, and produced as a means of testing for water pollution. Actually, they're now only used as an aquarium novelty. The company that produced them, Glowfish, fluorescent fish, actually, if you look on their website, they do think about some of the ethical principles involved in just generating a modified species just for fun, in terms of environmental safety, the humane treatment of the fish. But they are ultimately, they're just an aquarium novelty. Cupid and Diana, produced in 1999, they're sheep that express the human gene serum albumin. It's a blood protein that has tremendous medical uses. That means we could take these sheep, take the blood that they make, and they're making a human gene. So we don't need then to go taking non-human plasma, serum albumin, and using that medically. We're actually using human serum albumin. It just happens to be produced by sheep. Spider goat looks like a goat. It simply expresses in its milk the protein that goes to making spider webs, which is it has incredible physical properties. So here we have a goat that's making something that could produce new materials that we might use. Oncomouse is a genetically modified mouse that is destined always to produce tumours. It's used in cancer research. So you know that this mouse, unless it is treated with a successful anti-cancer agent, will go on to develop a cancer. Is that right for the mouse? And what right have we got to use it? Raise the questions. We might talk about this in the discussion afterwards. And even more emotive, a few years ago, monkeys were generated which we knew were going to go on to develop Huntington's disease. The fact that they look green in here is simply because the marker protein is that same green fluorescent protein. But they're genetically modified to have the mutation, that is a duplication of a region in front of a gene that goes on to modify the production of a protein called Huntington, which leads to that debilitating inherited disease, Huntington's disease. And these monkeys now are destined to go on and do that. So those are some examples, and you might think of why bother doing each one of those. What about the curiosity question? If we could insert DNA into animals, or human DNA into animals such as chimpanzees, just to see if we can make them more like ourselves, it might help us to define what makes a human. It would be fascinating to know, but apart from the fascination, why would we want to do this? I think for the curiosity. You might find those who might want to do it to create a new species, if you like, semi-human servants that could be used, subhumans. What is the end point of such research? And we need to bear that in mind as we do it. Let me raise you another question that's related to that. We're very close to sequencing the Neanderthal genome based on DNA extracted from Neanderthal bones that are tens of thousands of years old. It's not a complete genome. I think at the moment something like 65% of the Neanderthal genome is completed. And science fiction scenario, what if we could reconstruct, not just on paper, the Neanderthal genome, but if we could actually make it chemically? And what would we do with that? I think it's fascinating to know the Neanderthal genome, but if we could chemically make that and put that into a cell, what would we do? Or should we do that experiment? It would be fascinating to recreate Neanderthals, wouldn't it? To find out just what they were like. Why did they die? What properties did they have? What was their consciousness? If we did it, where would that Neanderthal be raised? Would that Neanderthal fetus be raised in a human womb or in a gorilla's womb? We're making value judgments in what is a Neanderthal as we do that. I'm personally quite convinced we should never do the experiment, but I fear that within the science, within the future, that there's no way at the moment we could do that. But in 100 years' time, I don't know, maybe it might be possible. Another medical benefit, of course, for doing transgenic modification is to produce organs for transplantation. There is a tremendous shortage of human organs for transplantation. There are many, many more people waiting for kidneys and livers and hearts than there are available. And of course the sad thing is that for every person who receives a transplant, of course somebody else has died. What if we could raise human organs within other animals? So the whole business of xenotransplantation. People don't want to take a pig heart and put it in a human, but what if we could humanize that heart? So there are pigs that have been raised that have human genes inserted within that. So that means you could take the pig heart and transfer it into the human, and there would be less signs of rejection. Are we meddling with nature with doing it? Are we simply using pigs as a means of plundering them for their organs? It's the yuck factor, again, comes into this in part, whereas, as Cheryl pointed out, when heart transplants started, people said you can't go around walking with somebody else's heart within you. That's horrible. It sounds equally horrible to us to say you're walking around with a pig's heart in you. Is there a yuck factor? Is there a real reason why we shouldn't do that? There are some very good scientific reasons why we shouldn't do it. There may be the possibility of diseases crossing species. And there's been the possibility of things called PERVs have been suggested. That's porcine, to the pig, endogenous retroviruses. Maybe within the pig, there may be viruses that we never get exposed to because they're actually confined and hidden within the pig genome, and the pig hearts put within us could expose that, and we might get a new round of fatal diseases. There is also the question, of course, that pigs are different from humans. There are different rates of growth, there are different blood pressures, their physiology is not the same as ours, even though there may be many, many similarities. But in terms of the pig, if you did it, I go back again, if it looks like a pig and acts like a pig, we call it a pig. There may be no harm to the animal, but there is the question of then what do we do with the organ that's raised in that animal. There's the question, too, of raising chimeras. And this will touch on things that will come up later on with what we can do with embryonic stem cells. And we can start to mix them together. Not making chimeric cells, but by making chimeric species, of mixing in the embryo some cells that come from one species and some that come from another. And again, this could be useful for generating organ donors. It could be used for generating species for testing drugs and for investigating a disease process. So you have something that can be generated called a guinea. And a guinea is simply by mixing in the embryo cells that come from a goat with those that come from a sheep. And it has characteristics that are halfway between a goat and a sheep. What about the process of growing a human heart in the laboratory, if it was technically possible? I've already mentioned the possibility of growing a human heart in another animal. If the process of the animal isn't harmed, is there really a problem? But let me raise you what may sound like a silly scenario, but just to raise it. We could have a chimera produced by injecting human cells into a mouse embryo. And then that embryo, that the mouse would have some human cells and some mouse cells. And it would probably look like a mouse, especially if it's only a very small number of human cells injected with that. Some of those cells could go on to make the germ cell of that mouse. So some of the germ cells could be human germ cells, and some could be mouse germ cells. So you could have the Frankenstein type scenario where two of these chimeric mice then breed together, and the germ cell, the human germ cell from one mouse joins with a human germ cell from another mouse, and you actually get a human generated which has two mice as parents. That sounds horrible and it sounds incomprehensible, but that technically could be a possibility. Let me move past that. So transgenic therapy has potential medical therapy, but it raises lots of questions. It raises questions of creating new life forms, long-term effects possibly on health and the environment. It involves questions of blending of non-human animal and human DNA and characteristics, and it could have unintended both personal, social and cultural consequences. So on the basis of that, you might say, well, let's stop doing it. There's too many questions to ask. Francis Collins said this, and I think this is actually very profound. He said that to say that genetic engineering is unacceptable across the board because of its potential for creating some ethical dilemmas is the most unethical stance of all. It's to basically say, here is a powerful approach which could alleviate some human suffering, but we're not going to do it because we're worried about the misuses that might occur. I find that completely unacceptable from every possible point of view. He's not saying laissez-faire, let's do everything, but he's also saying that we have a technology here, we're not going to use it because we're a little bit worried about it, is also an unethical and an immoral stance. Take the opposite view of this. Somebody called Jeremy Rifkin would take the view that actually we shouldn't be correcting human diseases, particularly genetic diseases. That it's the responsibility of those who have those sorts of problems to pass them on to the next generation. We shouldn't make value judgments about what is normal and what is abnormal, and we should maintain what we might call a rich a reservoir of genetic diversity. They're not mistakes, but options, some of which may become opportunities. Eliminating so-called bad genes risks depleting the gene pool and limiting future evolutionary options. There's also this scenario that we need to discuss about what is special about the human species as opposed to other species, as we do maybe genetic modification on mice or even monkeys. Some people have accused us of speciesism. Right? So whereas we might have sexism or ageism that says we discriminate again on people by virtue of their gender or their age, which of course there are laws for that make sure we can't do that, and we all know that we shouldn't. We discriminate on the basis of species, so that a mouse has different rights to a human. Why is that? So speciesism, as Peter Singer is one of the main advocates of this, is the idea that being human is a good enough reason for human animals to have greater moral rights than non-human animals. a prejudice or bias in favour of the interests of members of one's own species against those of members of other species. We are, most of us, speciesist. We would do things to animals that we wouldn't do to humans. It has to be said that most people object to this sort of a language, and putting it in the same context as racism or sexism or ageism just really doesn't seem appropriate or fair at all. Quickly, let's look at some GM applications. I've got lots more slides I could show you, but I think we'll cut short on this. Within the realm of pharmaceuticals, lots of things that can be done. Insulin used to be obtained from animal cadavers. And of course, some people had intolerance to pig insulin. Insulin is now made from E. coli. Human insulin made in a bacterium. Human growth hormone, again, used to be extracted from cadavers. It isn't anymore. It's made as a genetically modified product. Herceptin is one of the drugs used for treating breast cancer. Is a human antibody. It's made by recombinant technology. Interferon, used for treating many viral diseases, is made by recombinant DNA technology. So there are some fantastic advances in the whole areas of pharmaceuticals. In agriculture, with genetically modified crops, which are not used in this country at all. There are no commercial growth of genetically modified crops in this country, which is, I think, rather strange. But here's some examples. Golden rice is genetically modified rice that produces vitamin A. That means there's a natural vitamin supplement for use in parts of the world where there might be deficiencies in that. You can make rice that's resistant to drought, or rice that can tolerate all sorts of other different environmental problems as a reservoir of food. Many agricultural crops are made to be resistant to pesticide. That means that herbicides can be used in a more controlled fashion. Of course, there are risks involved in any of these in terms of containment. Could we get superweeds? There's questions of biodiversity. There's questions of toxicity and food safety. And every time these have been examined by the scientific community, it's demonstrated that actually there are very few risks at all. If any of these escape from the confines and get out into the wild, actually they're pathetic organisms, usually get outgrown by anything else that's there, and that there have been no instances of real demonstrated toxicity or food safety. It has been argued in terms of GM crops, in terms of the risk to biodiversity, there is a greater risk from garden centres. These places that are bringing in all sorts of exotic plant species from the rest of the world that shouldn't be here in this country, that threaten our biodiversity. But yet the public opinion matters. In Britain, 43% of people, we're told, think that GM food is very or fairly harmful. Now, whether they're right or they're wrong, that's what they think. In the EU, 56% think that it is dangerous. And you actually contrast that with the herbal remedies, which are usually pharmaceutically untested. 4% of people think that herbal remedies are very unsafe. Actually, herbal remedies, because they're untested, are often far less safe than many of the well-tested standard pharmaceutical agents. And within this country as well, there is a tremendous influence of the organic lobby. That those who think that food should be grown without recourse to any chemical intervention at all. And of course the organic lobby would not accept any GM modified crops at all. Let me give you an example of this, where we're weighing maybe one good against another. This dates back from 2002, it comes from the BBC website, so it must be correct. This concerns Zambia, where the Zambian government forced food aid that was being taken to that country. They discovered that they were distributing genetically modified maize, despite the fact the Zambian government had a ban on GM maize. This was a food stock that was needed because of a tremendous drought at the time, and as a result of that, of course, people went hungry and they starved and they died out of a political correctness that said we don't want GM modified crops. I suggest that one of the reasons why Zambia didn't want GM-modified crops in that country actually was because they then couldn't export their GM maize back to this country, because then the organic lobby wouldn't want to buy it. So it was imposing some rather special Western fads on a very needy place where the genetic modification could have made a tremendous difference. Just in closing, to raise questions, and I'm going to stop here. What about human genetic enhancement? Is there a difference between genetic modification for curing a disease, or for treating a disease at least, and a genetic modification in order to make us do things that we can't do at the moment? Maybe we can have a gene that would enable us to detect electromagnetic radiation, a gene that would enable us to see in the ultraviolet, or to see in the infrared. All those things that were mentioned in talks yesterday that other species have. Wouldn't it be nice to enhance humans? And should we do that? Again, beware, in some cases, of the genetic fallacy. Would it be good to take, say, a gene that gave particular athletic prowess or musical ability, so as to have the sorts of child who would become the musical genius? But of course, beware the genetic fallacy. Having a gene that says that you've got some muscle pattern that means you're going to be an athlete is not sufficient. You actually have to do the training. And some of us who foolishly go running every morning and do the training don't have the gene anyway, and it's not going to make a blind bit of difference, except for maybe altering a little bit of my cardiovascular efficiency. So I'm going to stop there, and there's lots more I could say about genetic enhancement of humans, but should we do it? What are the risks? What are the benefits? What does it say about what we are to be human? And I'll leave it there, and thank you for your attention.
Thank you very much, Keith, for that great overview, and over to you. Yes, please. Yes.
Are not we in danger of scope creep, that is, expecting one outcome, maybe getting all or part of that outcome, but also getting very unwanted and very unexpected outcomes? I have things like eugenics in mind. Human enhancement, well, would I have, would I love to run faster? Yes, etc., etc. But are we not in danger down the line of actually having to face things which we didn't expect? And as a scientist, I'm sure you would agree that there are absolute huge number of things which we face, but we didn't expect them to face. If you see what I mean.
Yes, I do. And that's a very, very good question. And it comes to almost any area of science. There is risk involved. You don't know the outcome. If you did know the outcome, then it wouldn't be science. In fact, I think I would criticise much of our government's science policy is they actually want to know the answer before they'll fund the science. So there always is that element of risk at the end of it, a what if. In terms of, you mentioned genetic enhancement of humans, there are a tremendous number of risks involved in that. But there are other ways in which we are already enhancing humans by non-genetic means, which carry with them risks that we need to think about. So the athlete may take chemical compounds to stimulate their performance. Students may well take mind-altering drugs that affect in the short term their ability to remember. And all of those things have long-term knock-ons. For the athlete who's abusing their body, they may well die of other diseases at an early age. For the student who's abusing drugs because either it feels good or it helps them to remember something, what's the net effect on their cognition, their psychological makeup 5-10 years down the line? And it's exactly the same with genetic modification, only it's a permanent put into the genetic stock.
So isn't it more fundamental? And what I was alluding to is the societal impact of it, like eugenics, in the sense that if we can say, well, this person is actually better in some sense, well, aren't we all, don't we all want better things? Then, we might be in great danger of going down the line, as Nazis went. well, gypsies are second class, these are second class. We don't really need them. They are actually wars. And, it's a common sense, isn't it? We should go for better and we should not go for the wars option.
Yes, I think, to be fair, at the moment, that is a very long way off before we would get to that sort of thing. But you could envisage a situation where there were two classes of humans. There were genetically enhanced and the ordinary humans. And what differences would that make? In terms of our value, it should make no difference at all. That we're all equally special, equally human, and have equal dignity. But in practice, it would then start to make differences. There were those who could be the intelligentsia, not by virtue of, if you like, the lottery, the genetic lottery that is dished out to you at birth, or the environment in which you're brought up in, but by virtue of the cash that had been put in to altering your particular embryo so that you were endowed with particular genes. And I think that comes with real problems.
Please, yes.
About 10 or 15 years ago, there was a lot of hope that gene therapy would be effective in single mutation diseases like cystic fibrosis, adenosine deaminase deficiency, and muscular dystrophy and so on. Have any of those hopes been realised in practice? And if so, sort of what are the outcomes of trying it?
To a very limited extent, the answer is yes. Some of those have been. There have been, particularly for adenosine deaminase deficiency, experiments done in both France and in England, which have been shown to work. Now the problem comes is that in many instances those genes have been transferred using viral vectors, and the virus inserts the gene into the genome, and you can't determine where it's going to go. So in many instances, as well as correcting the deficiency, and the knock-on for that is that later on down the line, is that patient then suffer from cancer, because the gene had interrupted a cancer-forming gene and a way in which that was controlled. So there are examples where it has been used. There have been great toxicities as a result of some of them. And I have to say that in some instances, there's been a tremendous amount of overhype of what can be achieved. Cystic fibrosis is still exactly the same as it was. You can look back at 20 years ago when that gene, the cystic fibrosis gene, was discovered, and the hype that went with that. This will lead to all sorts of new therapies and treatments and eradication of that mutation. And to be honest, some of it was well-intentioned hype. Some of it, I have to say that scientists are human as much as anybody else are, and that a scientist hyping up his discovery is likely to increase the chance of some form of funding coming through, and it's a game which we all play with the grant awarding bodies, of having to give some justification as to why they should fund this research project rather than that research project, and that's often not done on the basis of this is exciting science, but this will be a benefit to society. And sometimes those things are put in the general media, not just in the scientific press, and they get overhyped, and for good reasons or bad, and that sometimes it doesn't live up to the promise.
Question back there, yes. Please, yes.
Oh, thank you. Mm-hmm. Well, first of all, thank you. It's exciting and interesting talk with lots and lots of examples which were varying from inspiring to challenging. And that's where I began to question whether maybe
the God of Thunder, is...
whether maybe there is a warning here about the danger of absolutes. And you segued really from techniques that are current and possible and are happening to those that are in the realms of science fiction. And I thought you could have done this with many other modern techniques. You could have talked about grafting or prosthetics and had exactly the same sort of range of things that there are very few people who wouldn't say, oh, it's great if you can have an artificial hand, to those who worried a lot about artificial enhancement, prosthetics. And I wondered if a presentation such as yours, by blurring possibly the distinction between what is happening and what at the moment is fiction or fantasy, though I totally accept that what's fictional fantasy now becomes possible,
Goes back to that.
encourages the gut yuck reaction, and perhaps drives people towards absolutes and precludes a more measured response.
Yes, thank you for that. That is quite important. And part of the reason why I presented in this way, because I don't think there are absolutes in this. I don't think you can lay down a law that says you shall do this and you shan't do that. I think each one has to be weighed up on its own individual merits, the strengths and the weaknesses of that particular thing. Many of the ones we look back on are trivial, they're fun, there's no real ethical questions involved in them, but the techniques that have been developed from that lead through to other ones that could lead to nightmare scenarios in the future. And we need to be aware of those before they come and weigh up each occurrence in advance. Most genetically modified things, apart from the very trivial, have to go to ethical committees. And so I want to genetically modify E. coli in the laboratory. I will do that, and my lab notebook has to record that I've done it. And it has to be proper containment. If you want to do any other form of genetic modification, it usually has to go to an ethical committee that will say whether it fits within the legal framework, whether there's a need to do it, what the containment is, what the outcomes might be. So we do have some form of framework in this country, at least for deciding what might be appropriate on a case-by-case basis. I think I was suggesting that we don't want to lay down the law and say, here are the shutters, we shouldn't go any further, but that we need to be very careful as we move through. Yeah.
Yes, please.
I think you've been exceedingly cynical about Zambia and its rejection of GE corn, particularly from the perspective that they actually knew the situation and they went obviously through an ethical and moral decision-making process to decide whether or not to take that particular form of aid. And I think, just to dismiss it as they wanted to sell their crops into Europe is being absolutely cynical about a major moral decision that was made.
Thank you for that.
And furthermore, we could ask who's recovered better from that drought? Those that took that kind of GE corn or Zambia? And I think Zambia might come out better.
It may be, but I also wanted to raise that, and I didn't make it very clear what I said, so thank you for raising that again. It's another question of weighing the good with the maybe the harm that might be done. Which is the better to take the short-term policy, or maybe a long-term policy, that says we don't want GM food, or weigh that against the need for people to be fed.
But the further point is that the exporting nation wasn't allowing that, but they were putting it into their aid.
Yes, I couldn't comment on that.
I think we have to be a little bit clearer about this.
Certainly within the US, most maize that is made is grown highly...
But not for human consumption directly.
Yes, I think that's it.
But this was for human consumption. There's a major moral difference in that, ethical difference.
I'm not actually sure that there is.
Well, they make the distinction in the States. But for a poor, starving African country, they can eat it. There is a major issue there.
There is, but it's still the issue of which is better for people to starve or not to starve. I don't necessarily throw this out because I have an answer to it, but it raises the ethical question of doing good, doing harm, and balancing those two together. Now there is if you take the line that GM maize is evil and should never be consumed by humans, then clearly you're going to say that it shouldn't be consumed by humans in this country or the States or Zambia.
No, that's not my line that I'm taking. The line I am taking is that the exporting nation says that it's not for human consumption in the first instance. They then are using that as aid to a country in an incredibly difficult situation. I think we need to be respectful of those decisions that were made not from a comfortable situation, but actually from a situation of incredible pain, and yet they would make choices. And I think we need to be careful and actually look at the choices and the reasons that made it, and not dismiss it because they want the export.
Thank you, and that actually raises the whole question of autonomy within this as well, in making those sorts of ethical decisions. It is, it would be the Zambians' right to decide for themselves whether they want that and not to be imposed from outside.
We have a question over here, thank you. Yes.
Probably around the same issues. It's the question of science, corporations, money. Like when we enter into this dialogue, you said yourself about you have to present your papers if you want grants. You have to present them in a certain way. And the same for this whole question. I think it leads to, there has to be some form of ethical reflection from outside the scientific community also.
Yes.
And also the same outside religious community. I think.
Yes. Yes, I agree with you. There has to be reflection, not just the scientists looking at it, because they often can't see the wood for the trees. And there has to be reflection amongst people of all faiths and all communities and all societies looking at this in together. The one thing I would say, a slight issue, you hinted at, is the question of commercial value within these sorts of things. And it's used as an argument sometimes against GM, where there's the big companies, the Monsantos and whatever, that are after making money, which is why they want to do this. That's what the world is like. I mean, all of industry out there is not actually out there for the public benefit. The pharmaceutical industry, for instance, may produce lots of drugs that have fantastic use for curing diseases, but the pharmaceutical industry, at bottom line, is after making money in the same way as with any industry, whether they're making radios or transistors or computers or whatever, they're making it to make money because the public want it. So I don't think we can necessarily make an evil out of making money out of these things. That's just the very nature of the society in which we live that fits with other things.
I agree with that, but I think there can be a danger that then it becomes tied to other things.
Oh yes.
I think that's where a danger arises, when you have to accept something in order to get something else.
Yes, I mean, and the GM crops is particularly, and I think that's now been dealt with, but there was in the early days the possibility that you went to a particular company to get your GM modified maize, and you'd have to then go back to the same company time after time after time. You'd be tied in long term into their economy rather than making a decision as you go. That I think is now dealt with.
Okay, so we got a lot of hands here. I think we'll go right to the back and then forward again. Yeah.
Just to pick up on that point about not just the scientific community looking at it, and from a social perspective of, I think the distinction is it's a difference between improving somebody's quality of life and undermining human dignity. I think that's the distinction that has to be made. The other thing is we live in a society that is constantly trying to balance out the differences in terms of, say, education, the removal of a class-based education system, and the removal of class-based in general. If it's going to be a case of the person with the most money is going to be able to enhance themselves the most, are we not embodying a class-based system? And just as a pure side point, are we going to wind up with a genetic Olympics, whereby somebody with 3 days can run 100 metres?
I agree with you completely, but that's why I wanted to raise human enhancement at the end. In many ways, I think it's a no-brainer. It goes contrary to everything that we strive for in terms of equality and fairness and justice. But nonetheless, the concept is out there, and we need to grapple with it. But I take your points exactly with that. Thank you.
Yes, please.
And could I briefly point out, just simply because it happens, it didn't arise yet this week, the GM case in Britain, and I think it's much simpler to look at it in one country. In the case of Britain, there's a perfect example of a very big gap between the public perception of science, as you pointed out, and the consensus of scientists. And I don't mean this in a patronising way at all. In fact, quite the opposite. But I think, governments, as Stephen was pointing out, they're democratically elected. It's their responsibility to reflect what the majority think. But it's not fair. Is it fair to ask the majority to take a decision on something or to have an opinion on something that really they're not given the information on? And that's the one issue that I want to mention that hasn't actually come up this week, is the whole thing about science education, that we live in a world where science is now such a part of the world that no matter what religion you are, no matter where you're coming from, basic scientific literacy in the sense of being able to read and write, literally as basic as that, is becoming almost completely indispensable, I think.
I totally agree. I mean, thank you. But I don't, apart from encouraging science teachers, and there are a few here, what a fantastic job they do and they're required to do in order to make sure that the kids know the basics of science and understand, or are given the tools to be able to understand the new problems as they come, new technologies that they emerge. But we have to live in a world where the vast majority of people around us just have no understanding of the basis of the science, make their decision on the basis of what they read in the Daily Mirror and the Daily Mail in this country. They may be wrong in their assumptions, in some of the prejudices they come with, but we have to live with that in the first instance and maybe educate beyond that in the longer term. If for maybe even for the wrong reasons somebody says they don't want GM, we need, I think, to respect that view at the same time as educating you, and that is difficult. There is sometimes the nanny state can go either way and say we know what's best for you. They may not know what's best, but do they have the right to impose that on people? That's a difficult issue.
And many people still think that the sun goes around the earth, I think, don't they? I mean, when you do these polls, actually, it's amazing, So that's sort of, it seems fairly basic compared to something complex like GM. So I think there is a huge communication problem. Yes, Hilary.
I just wanted to pick up again on the issue of the Terminator seeds with GM, because you say that's now been rectified, and public opinion presumably played a large part in that.
Yes.
But it struck me that actually it's a potential danger, isn't it, that the commercial aspect of it won over first, and almost without people realising, that unethical commercial agenda was prioritised, and then the reflection on the social justice aspects came to bear. And it struck me as, is that the paradigm for all these new technological developments? And if so, isn't there a sort of fundamental principle that the ethical aspects should be considered first?
They should be considered first, and that I think goes back to one of the quotes I had from Steve Jones at the beginning, is often we're trying to play ethical catch-up. I'm heartened that even though the ethical considerations were in that case given later, nonetheless they won. The pressure put on the companies with these things like Terminator genes was such that the issue was dealt with.
It begs the question, what perhaps more subtle aspects does commercial interest win over, where actually public awareness, people don't find out about, or they don't know about, or there's not perhaps such a stark outcome, if you like?
Yes, I don't know. One could be suspicious and cynical of the whole commercial enterprise and suspect that there's reds under the beds, and I don't know.
I mean, if it wasn't the motivation of pharmaceutical companies to make money for their shareholders, we'd all be dead. It's that simple. So I mean, just to put a little point in that direction, just to balance the discussion off a little bit. I mean, most of us would be that anyway. So, the profit motive isn't entirely evil. I mean, it can actually lead to some good things. So, and that's what, I guess, the capitalist system, but let's not. We'll go there and then here. Yes, thank you. Yes.
Clearly, the science that gets funded within a country is determined somewhat by ethical committees within that country. Are there international ethical committees that look at sort of a global perspective of where ethics is at?
I hesitate to ask. I mean, maybe Cheryl, do you know better on that in terms of international?
There's certainly, for medical ethics, there's a global organisation. I'm not sure about for agricultural or medicinal pharmaceuticals.
There's one for physics.
Oh, sorry, there's one for
physics in particular, mainly because of it.
Oh yeah, but I'm not sure whether there is one for things like agricultural use or animal use of genetically modified organisms.
So the supplementary question, I guess, is that you could have countries in which the ethics is not really looked at in much detail, and you could have commercial or other scientists going to that country and being set up in
Oh yes. I've talked with people. The department I'm in is having a new building constructed, and we've employed international consultants in order to get that building up and designed and furbished. And talking to one of their staff who flies transatlantic all the time, he said that when he's flying to this country, he meets Americans coming over here because they want to do stem cell research. And when he flies back to the States, he finds English people going over there because they want to do animal experiments and found it over here. Right? So even within the US, Great Britain, scientists move around according to what they want, what they want to do. Within the question of, say, the scenario of cloning a human, right? It's banned in most civilized countries. But the facilities there, and maybe a bit of luck in a rogue nation, where somebody wants to do this for the sake of their own ego, and it might happen. And so there aren't international laws that every nation would sign up to.
There was another. Yes, please. Yes.
A quick question: is it right that the UK tends to be overall sort of further or pushing the boundaries more in terms of genetic modification and sort of doing new experimentation? And if so, why do we tend to be pushing the boundaries more here?
I don't think we, in terms of research, we might be. In terms of application, I don't think we push any more than any other nation. I think we have a very good research base in this country. We have the technology, we have the people with the skills. That's the reason why things happen here. Not because the laws are any more lax in this country.
Okay, so there's no particular sort of ideology that promotes
so.
more.
Yeah.
The other, can I answer very
Go.
In terms of sort of getting it for I suppose, as general lay people, let's say living in the UK, just to understand the process a bit more in terms of how things, you talked about ethical committees, etc. How does the process work, just sort of basically, and also how, if we, the general public, are going to get involved in any more in the ethical decision making and influencing things, at what stage do we get involved to engage in the debate?
Right, now for the average lay person, to be honest, I don't think I have an answer to that question. So if you have a bee in your bonnet as a lay person, how do you make representation of that to the right ethical committees? I don't think I have an answer there. We could certainly do with educating people as to what processes have gone through before anything is let loose on the general public as a therapy, any form of medical treatment, and the kinds of controls that have to go into that, the safeguards in making it, the risks, the health and safety, all those sorts of things. We could make those sorts of things much more obvious to the general public, but my guess is that they probably yawn in the way that most of us get incredibly frustrated in the science lab and filling in paperwork to do with health and safety half the time instead of getting on and doing the work.
So it's really just sort of the final stage when it gets to sort of parliament stage, a legislation stage where people can really get up and do something.
Yes, yeah. So you can write to your MP, who can raise things in Parliament, and obviously within the recent past, the whole human embryology bill, that I guess others will say more about later this afternoon, was a real case in point where I think again the public probably had misconceptions about what was being proposed in aspects of that bill, but nonetheless it was something that was there on the agenda and that people could feed into.
I mean, it might be worth adding that a lot of places would have, the ethical committee or the welfare committee in place, and they deliberately have, if you like, lay members as part of that committee from the local community, and certainly at the Abraham Institute, where I've been working these past years, that is the case, so that is part of the policy of the committee, is to incorporate people from the non-scientists onto the committee, so that they're also part of the decision-making process, so that happens as well. We're running a little on time, but yes, maybe time for one last question, and then, yeah, okay, yes, yeah, then we should, yeah.
looking at the record of the Parliament, or not just of this country, but any other legislative body, do we have really grounds for faith in their ability of an ordinary, regular MP to understand all the difficulties? What I have particularly in mind, as an example only, I'm sure there will be probably other examples, the Human Tissue Act. Obviously it has been enacted. Now we have to comply with it, and obviously you know much better all the difficulties of complying with it, all the intricacies of when it applies, it doesn't apply. Can we expect Parliament? Can we expect the 600 MPs, who are most of them are not scientifically or medically qualified, to understand all the details and enact for us what we can do and what we cannot?
Thank you. I think that's an extremely important point. Is that most of our MPs are not scientifically literate. They may be intelligent people. A vast majority of them will have done as their academic discipline a law-based or journalism, but very few come from a scientific or a medical background. There are those that are there, and maybe that again says how we need to be educating people right across the board, so that our MPs, even though they come from maybe law, as a lot of them do, that they have a strong scientific background that says that they may not have done it to university, but they're equipped with some of the skills to be able to deal with some of the problems. And even if they're not equipped, that their advisors should be equipped. But we are, I think, a lot of the time putting these decisions in the hands of politicians who are very well-meaning, but maybe don't have personally the skills to be able to deal with them. So thank you. I think that is very important.
Okay, I think we'll stop there. Although actually, we might just feel like staying here for a bit. But I know perhaps people want to stretch their legs. And so, first of all, let's thank Keith for a really interesting talk. So thank you very much, Keith.